Welcome, everyone, to the 2024 Graduate Research Seminar. My name is Tracy Johnson, and I'm a supervisory physical scientist in the NIJ Office of Investigative and Forensic Science. Here during Forensic Science Week, we are going to have a graduate research symposium. This event provides a platform for graduate students and early career scientists to present their research to a broad audience. Now I would like to introduce our first speaker, Remy Nguyen from the University of California, Santa Cruz, presenting a computational method for genetic identification from low coverage sequencing data. Take it away, Remy.
Thank you, Tracy. Hi, everyone. Thank you for coming to the Forensic Science Graduate Research Symposium today. My name is Remy Nguyen, and I am a PhD candidate in the Paleogenomics Lab at UC Santa Cruz. A main focus of my research work has been to make the genetic analysis of forensic samples more robust and reliable, despite the fact that many of these samples are difficult to characterize using standard assays. And my talk today will go over my current progress on that effort, which is the development of a computational method for genetic identification from low coverage sequencing data. First, I would like to go over some background. Genetic identification, also known as DNA profiling, is a process that involves identifying an individual based on their DNA sample by looking at the unique variations and patterns in their genome. This technique was first developed in the 1980s and has since been used in various contexts, including in the justice system for convicting or exonerating a suspect, identifying missing persons or disaster victims, or in immigration and paternity testing to establish relationships between relatives, as well as in genealogy to understand family history. Traditionally, short tandem repeats, or STRs, are used as a marker system for genetic identification in forensics. These are microsatellites with repeat units ranging from 2 to 7 base pairs in length, with the lengths of the repeats varying across individuals, making this type of genomic variation especially valuable in identity analysis. A routine practice in forensic investigation is to compare the STR profile of a crime scene evidence with that of a suspect in order to calculate the likelihood of a match. And the proven effectiveness of this marker system has led to the establishment of 20 standard STR loci, now commonly used in the CODIS database in the US. A widely used method for generating an STR profile is through PCR, or polymerase chain reaction, that can produce millions of copies of DNA spanning the regions of interest. However, this approach has many disadvantages. One of them is allele dropout due to the stochastic effect of PCR amplification, leading to false homozygosity diagnosis. Furthermore, polymerase slippage is a common problem, which results in fragments of various lengths that show up as theta peaks in the electrophoresis signals and can complicate the interpretation of the data. And finally, in extremely degraded samples, or samples that have very little quantities of DNA, fragments cannot be amplified at all with PCR, and this method would simply fail. Because of these drawbacks, in recent years, single nucleotide polymorphisms, or SNPs, have been explored as an alternative to the STR system. These variants can be genotyped directly from the sequence data or through microarrays. Genome-wide SNPs can also be used to detect shared regions of common ancestry along the DNA sequences of two people. These regions are known as being identical by descent, or IBD. Because recombination over generations can break up IBD regions into smaller segments, people who are closely related will share longer stretches of identical by descent DNA. And in contrast, more distant relatives will share shorter IBD segments. IBD detection, therefore, serves as the basis for the relative finding feature that are included in many direct-to-consumer genetic testing products like those from 23andMe or Ancestry DNA. And because of this, we can think of cell identification as a special case of relative finding, in which we expect the compared genotypes profiles to be identical across nearly all sites. So using this logic, I have developed a method for inferring the relationship between two forensic DNA samples, taking advantage of information from low coverage sequencing data and SNP genotype data. This method is called IBD-GEM, and in a nutshell, it is a computer program that compares genotype data from a known individual to sequence data from an unknown forensic sample, which are shown respectively by the blue and the tan ribbons in the schematic. In addition, the program takes in genotypes from a set of reference individuals to estimate the background genetic diversity in the population, and these are shown in gray here. At each variant, for example, from SNP1 to SNP5, IBD-GEM uses allele count data from the sequence reads, as well as the genotype profile of the query individual to compute the probability of observing the allele counts given the test sample and the query genotype profile share 0, 1, or 2 identical by the same chromosomes. For the rest of the talk, I will refer to these hypotheses as IBD0, IBD1, and IBD2 models. For identification purposes, we are interested in whether these samples share 0 chromosome IBD, which means that they come from different individuals, or whether they share 2 chromosomes IBD, which means that the samples come from the same person. These two models are highlighted in red here. After IBD-GEM has generated likelihood results for these models, we can compute the likelihood ratio of IBD2 over IBD0. And highly positive log likelihood ratios means that the sequence and the genotype data likely come from the same person, whereas a negative number indicate that the two samples are likely unrelated. We can aggregate these likelihood ratios over a genomic region. For example, taking the product of the ratio from SNP1 to SNP5 to increase the discriminatory power between the two models. And next, I want to evaluate the method's performance. And so I use DNA sequence and genotype data from the GBR and the LWK panels in the 1000 genomes dataset. This is a publicly available dataset that contains genetic data for more than 3,000 individuals from various populations around the world. GBR is the population group of European ancestry, and LWK is of African ancestry. Here, same individual comparisons represent positive controls in which all regions across the genome are expected to be IBD2, and different individual comparisons represent negative controls in which all regions should be IBD0. And in this experiment, I compared the genotype of each individual against either their own sequence data or the sequence data of another random individual that I had downsampled to various genomics coverages from 2x all the way to 0.01x. The two plots here show the distribution of mean log likelihood ratios for comparing each panel at those coverages. And as you can see, even at the lowest coverage of 0.01x, IBD-GEM is still able to differentiate between positive and negative comparisons in both the GBR and LWK panels. And because IBD-GEM requires a reference panel to model the background genetic diversity, we next wanted to know if the choice of our reference panel could meaningfully affect the likelihood result that it generates. And in the last experiments, I used genotype from all unrelated individuals in the 1000 genomes as my reference panel. And here to test the sensitivity of IBD-GEM to the genotypes from specific populations, I instead used genotypes from 5 different populations to model the background. And in particular, it's the African, American, East Asian, South Asian, and European populations, and then repeat the comparisons for the GBR and the LWK panels. We expect that doing so would not have a big effect on the resulting likelihoods, because in general, humans have very low cross-population genetic structure. And the likelihood ratio distribution shown here confirm this expectation by suggesting that even using the reference genotypes from a population of a different ancestry, for example, using African genotypes as background for running comparisons on European individuals, positive and negative comparisons are still easily distinguishable from each other. And after testing the method on the 1,000 genomes data, I wanted to evaluate the method on samples that are more similar in nature to those from actual forensic sources. And we know that hair is a ubiquitous sample type in forensics, and DNA from rootless hairs are especially difficult to work with since they are highly fragmented. So we put together a panel of head and pubic hairs, as well as saliva samples collected from 8 individuals. And Josh, a postdoc from our lab, trimmed the roots from the hairs and extracted the DNA from both the hair and the saliva samples. The plot on top shows the coverage distribution of the 8 hairs on chromosome 1, and the bottom plot shows the coverage distribution for the corresponding saliva samples on the bottom. And the distributions for the hairs vary quite a bit, but in general, they are all lower than the saliva samples, with 3 hairs having less than 1x mean coverage. We also generate genotype array data from the saliva libraries, which would then be used to compare against the sequence data from the hairs. And for this analysis, similar to the 1,000 genomes experiment, I ran 1 positive and 1 negative comparison on each individual in the hair panel. The box plots show the distribution of likelihood ratios aggregated in 50 SNP windows across the genome for both positive in orange and negative comparison in blue, with lower coverage hair samples on the left and higher coverage samples on the right. This suggests that IBGEM can accurately differentiate between positive and negative comparisons using very low coverage sequencing data from actual rootless hairs. And in addition to the main goal of identifying unknown samples, I also implemented an additional module called Hidden Gem that finds the most likely path of IBD state across the genome. And this module can therefore be used to infer the degree of relatedness between the compared individuals. The 2 bar graphs correspond to the results from comparing 2 individuals from the 1,000 genomes to their known relatives, with the genome-wide proportion of each IBD state shown for each comparison. We expect the parent-child relatives should be 100% IBD1 because the child inherits exactly 1 copy of the genome from each parent. Meanwhile, full siblings should show 25% IBD0, 50% IBD1, and 25% IBD2. And half siblings, grandparent, grandchild, and avuncular relationships should show 50% IBD0 and 50% IBD1. We know that variations in these numbers are expected due to a random nature of recombination, but in general, we can see that the proportions reported from Hidden Gem show reasonable concordance with the expected values, given the known degree of relatedness for each comparison. And next, we wanted to see if there is a good way to create a single, more useful metric for differentiating between positive and negative comparisons from IBDGEM for reporting purposes. To do this, we propose aggregating the likelihood ratios across each arm of each autosome. Then the chromosome arm with the minimum likelihood ratio would serve as a conservative metric for telling apart same and different individual. The plot here shows the chromosome arm statistics from comparing one individual from the 1,000 genomes to either their known relatives or to themselves. Each data point on the plot is the cumulative likelihood ratio over a specific chromosome arm, with a point in red corresponding to the arm where the likelihood ratio is the smallest. For comparison with known relatives, such as those in the top 4 plots, the red data points always fall well below 0, whereas for the self-comparison in the bottom, even the lowest chromosome arm value is still highly positive. This tells us that these statistics are clear and powerful enough to help us distinguish between actual self-identifications and from comparison with close relatives. And finally, for one of our most recent analysis, we expanded our hair panel so that it now includes 80 rootless hairs from 50 individuals, together with 50 corresponding saliva samples. The hairs represent a mix of both head and pubic hairs, with DNA sequence coverages ranging from 0.4 to 4.5x. We then evaluate IBD-GEM on this new dataset, running positive and negative comparisons on each hair sample. Here, the red points represent positive comparisons in which we compare the sequence data from each hair to its matching saliva genotype. Meanwhile, the blue points represent negative comparisons in which we compare each hair to the genotypes derived from the saliva DNA of 49 other individuals in the panel. And once again, we can see that there is a clear separation between positive and negative comparisons in terms of their chromosome arm likelihood statistics. And this distinction becomes greater as the coverage of the hair sample increases. And in conclusion, we have shown that our method, IBD-GEM, can make reliable identity detections using low-coverage DNA derived from single rootless hairs, which are a common type of samples in forensics. And additionally, with the Hidden Gem module, we can also identify genomic segments of relatedness and infer biological relationships between the compared individuals. My next steps for this project will be to improve the runtime and make the program more user-friendly by offering compatibility with standard file formats. I will also optimize the relatedness detection function so that the method can identify relatives beyond the third degree. And finally, I will continue evaluating the performance of IBD-GEM in the presence of genotype or sequencing errors, as well as sample mixtures.
Remy, now we'll spend the next few minutes discussing questions, and I will start with the following question. Do you have a scale of likelihood ratios, and what do they mean for IBD, and how does that relate to or not to other DNA likelihood ratios?
I have not compared the magnitude of likelihood results generated from IBD-GEM to the magnitude of the likelihood results generated by other methods. I have only, but that is a good question. I've only been testing, determining the determining whether the samples actually come from the same person or from different individual based within the test itself, and have not been doing any cross comparison between different methods. But yes, that would be a good consideration.
Thank you. So our next question is, do you think this method will also be useful for ancient and or degraded samples?
Yes, that is also a great question. So the hairs samples that I have tested here are modern hair samples. So I think they so the DNA in them are maybe fresher and not as degraded as we would expect in ancient samples. But I think in the future we also plan to try to artificially age the hair and also test the method used on that to see how it would fare on older sample as well.
Thank you for that. Our next question is, how far out in the family tree can these IBD likelihood ratios predict the degree of relatedness?
So we have only been able to determine relationships up to the third degree. So for example, first cousins and grandparents, grandchild, and uncle and niece or nephew. But I hope in the future to continue improving the relative detection feature so that we can detect more distant relatives using this method.
So I have a quick question. So I'm kind of going off script here in terms of the chat pod. But being familiar with sequencing data, how does IBD-GEM deal with heteroplasmy?
Yes, that is a really great question. I have not evaluate IBD-GEM using heteroplasmic data, but I expect that it will not alter the likelihood results too much, but that is a good consideration and test to perform in the future as well.
Thank you. So we've had a few more questions come in. I'll start with this one. How big does a reference panel have to be in order for IBD-GEM to make accurate identification decisions?
So I have tested IBD-GEM on a smaller subset of using a smaller subset of reference genotypes, and I found that even so, the 1,000 genome panels has thousands of individuals and have been using those as the reference panel, but I have using a subset of those panels as well, and I found that using only 50 individuals as the panel can offer pretty accurate results. So I believe it's because of the low genetic structure among populations that we can use a smaller panel as a reference.
All right, we have just a few more questions here. Have you tried using a smaller panel than the 1,000 genomes panel?
Yes, so I have subset the 1,000 for 1,000 genomes panel to a random 50 individual panel, and I got pretty accurate results as well. So I haven't tried going even smaller than that, but yes, I will test that in the future.
All right, thank you. So we have one last question, and then we're going to move on to our next presenter in the interest of time. So Remy, how does IBD-GEM take into account linkage disequilibrium between variable sites, which is known to exist in dense SNP panels like the 1,000 genomes?
Yes, I have included a linkage disequilibrium mode within the program so that as it calculates the likelihood for the background panel, it will compare the sequence data to the genotype data of each individual in the panel and taking the average as the likelihood for the IBD0 likelihood. So in that way, it will account for linkage across multiple SNPs instead of treating each SNP as independent variation.
That was, I think, an important closing statement to help the folks understand exactly how your software application is evaluating the data. So thank you very much for your presentation, and we're going to be moving on to our next presenter. So up next, we will hear from Leah Auchter from the University of Nevada, Reno. Leah, you can go ahead and start sharing your slides. Leah's talk will be about subadult sex estimation methods using pelvic metrics and geometric morphometrics. Wow, that's a mouthful. So the floor is yours, Leah. Thank you.
Hello, everyone. Hopefully you're seeing the full screen of my slides right now. Like Tracy said, my name is Leah Auchter, and I'm currently a PhD candidate in biological anthropology at the University of Nevada, Reno. Firstly, I wanted to quickly thank the NIJ and the Forensic Technology Center of Excellence for the invitation to present at this year's Graduate Research Symposium. I will be presenting just a small portion of my NIJ-funded research, which focuses on the ontogeny of the pelvis. So today, I will be discussing my initial results for the subadult skeletal sex estimation method using pelvic metrics and geometric morphometrics. The job of a forensic anthropologist is to generate an anthropological report to aid in the identification or analysis of skeletal remains within a forensic context, and can include skeletal inventory and taphonomic analysis, a skeletal age estimation, and trauma analysis. Additionally, one potential component of the forensic anthropological report includes the skeletal sex estimation, or the assessment of the downstream effects of sexual dimorphism on the size and shape of skeletal elements. Although there are multiple metric and morphological methods that have been validated for use in adult individuals, multiple problems have been encountered when attempting to develop methodologies for the skeletal analyses of subadult individuals. In this context, Subadult classifies those individuals that are still within stages of skeletal growth and development. The current lack of validated methods for subadult individuals adds to the difficulties of identifying unknown skeletal remains of young individuals. These problems include a small number of sample sizes, which are particularly affected by non-equal age distributions. The lack of sufficient sample sizes then limits the accuracy and prevents validation of methods. Additionally, historical or bioarchaeological subadult skeletal collections may have unknown chronological age and biological sex. Non-contemporary samples that may include demographic information have been shown to demonstrate secular changes in growth patterns, and therefore methods may not be applicable to contemporaneous cohorts that we would encounter within the forensic context. So there is a need for improved skeletal analyses and methodologies for subadult individuals. The use of computed tomography or CT scans has addressed many, if not all, of the problems that have been facing subadult skeletal analyses. The use of CT scans allows for the analysis of a contemporaneous population with known demographics, in situ measurements, and a large number of individuals. The individual skeletal elements can be selected, isolated, and reconstructed using a 3D surface model. Studies have demonstrated the reliability and accuracy of measurements across CT and dry bone such that they are comparable, and therefore methods developed using CT imagery can be applied to dry bone cases. For this current study, a skeletal maturation indicator was used as a selection criteria to include only individuals with a complete innominate. A complete innominate, or in this case, the left side of the pelvis, is indicated by active or complete fusion at the triradiate cartilage, which is the central portion of the innominate, meaning that all three independent elements are fusing together. The complete innominate was chosen based on suggestion from previous studies demonstrating that the individual elements lacked reliability when used for sex estimations. Additionally, a complete innominate allows the forensic practitioners to use published standardized measurements and removes the need to assess chronological age prior to applying the method. The only requirement for the proposed method is a complete innominate, which is easily visualized and does not require any additional assessments. CT scans were collected from the Subadult Virtual Anthropology database, which was originally sourced from the New Mexico Deceded Image Database, or NIMDID. Both of those databases are results of previously funded NIJ projects. NIMDID is a virtual repository of full-body CT scans collected from the medical examiner's office and includes individuals who died between 2010 and 2017 in New Mexico. The reported demographic information, such as biological sex assigned at birth and chronological age, were included with the anonymized CT scans provided by NIMDID. Therefore, the biological or skeletal sex in this study follows a binary model used by the medical examiner's office. The selection criteria resulted in a sample of 364 individuals, with 143 females and 221 males. Depending on the method, the sample size did change slightly. The age range produced from the selection criteria was from 8 to 20 years, with the majority of individuals 12 years and older, which can be seen in the age distribution plot shown here. Both a training and testing or holdout subset were generated from that total sample. All of the CT scans were processed, and the pelvic elements were isolated and segmented to produce the 3D surface models. Once the pelvic surface models were produced, virtual landmarks were placed on all of the left innominates when available using the Amira software. 34 pelvic landmarks were placed. Depending on the stage of fusion, that number does change. X, Y, and Z coordinates were extracted for each landmark and used for all of the following analyses. The two methods tested in the study for subadult sex estimation included standard inner landmark distances and geometric morphometric analysis. Discriminant function analyses were performed using the ILD metrics and also using the principal component scores from the GM analysis. All the statistical analyses and visualizations were generated using RStudio. And the DFA was used to estimate or predict the skeletal sex of the individuals. For the first method, a set of 11 standard two-dimensional ILDs defined in the forensic anthropology literature, which have been tested and validated for adult sex estimation, were applied to this subadult sample. Some definitions were slightly adjusted for use in a virtual sample and for subadult individuals. These images here show where those ILDs were measured on the CT scans. Results of the DFA using all of the 11 ILDs are shown here. The total correct classification accuracy using the holdout subset was 97% correct. The density plot shown here visualizes the separation between females in yellow and males in blue. Male individuals that were misclassified as females by the model are indicated with a solid line, and misclassified females are indicated with the dashed line. Both of the two misclassified individuals in this model fall within the overlap between the females and males. A second DFA model was produced using 8 stepwise selected ILD variables. All the variables used in this model additionally demonstrated high intra-observer reliability. The density plot here again shows the separation between the scores for females, again in yellow, and males in blue. 2 males were misclassified as females and are indicated by the solid lines. The total correct classification for the holdout subset, which functions as the validation method, again resulted in approximately a 97% accuracy. Some of the most important measurements used in the DFA are shown here, plotted against age. For both the maximum innominate height, which is in the top figure, and the ischial length, shown in the bottom figure, males and females have a separation of measurements throughout the age range, with males measuring larger than females. The second method explored in this study was a geometric morphometric, or GM, shape analysis. For the GM analyses, the software Morphologica was used. A wireframe was defined using 17 of the pelvic landmarks to provide an outline of the innominate shape. A Procrustes alignment was then performed to scale the size, shift the position, and rotate the orientation of each individual wireframe so that all individuals were aligned in a 3-dimensional coordinate space. A principal components analysis was then performed on the aligned wireframes with the adjusted landmark coordinates. All of the further analyses were performed using the principal component scores. Classification accuracy for the holdout subset, again, that validation method, using all of the PCs as predictor variables, was 96.5% correct. So here we are also seeing a high classification accuracy, just slightly lower than the ILD method. The density plot again shows the separation between females, again in yellow, and males in blue. Using this model, 3 individuals were misclassified, 1 male identified or indicated with the solid line, and 2 females indicated by the dashed lines. Again, they're all falling within that overlap between the males and females. Here's an example of the GM shape analysis output. So focusing on the area circled in red on the wireframe on the, I believe, left of your screen. This is the anterior portion of the innominate, which includes the pubis and the ischium. And we can see the elongation that occurs in the bottom image compared to the top image. As we move across that second principal component, or from the top of the graph where the green cross is plotted to the bottom of the graph and the red star, we are seeing the elongation of that anterior portion captured in the shape analysis. We can also see the females are clustering in the bottom half of the graph nearer that red star, and the male individuals are clustering at the top of the graph. So they're divided along that PC2. Summarizing some of these changes captured in the GM analysis, we can see that from the most female-like or most negative PCs to the most positive PCs or the most male-like innominate, there is an increase in the maximum innominate height, an increase in the minimum breadth of the innominate, a decrease in the lengths of that anterior portion of the innominate, which includes the minimum and maximum pubis lengths, and a decrease in the width of the greater sciatic notch. Most of these changes shown in the GM shape analysis were also reflected in the metric analyses. When looking at the specific performance of the 2 models in relation to the age of the individuals that were misclassified, we can see it is not consistently the younger individuals being misclassified, as we might have predicted based on previous studies that suggested sex estimation was unreliable in the young individuals. The stepwise selected ILD method misclassified a 14-year-old and a 20-year-old male, while the GM method misclassified a 12 and 16-year-old female and a 16-year-old male. These misclassifications represent the expected degree of human variation that exists in pelvic size and shape, and the misclassifications occur across the included age range, and both males and females are being misclassified. So to summarize these findings, the ILD analyses demonstrated slightly higher correct sex classifications compared to the GM. The misclassified individuals likely represent the degree of human variation that exists in the pelvis, as all of the individuals did fall within the overlap on the density plot and were not consistently of one age group. The most important ILDs for the correct sex classification were also visualized and captured in the shape changes across the PCs with the highest percent variance explained. These included the ischial measurements and maximum innominate height and pubis length. And again, the correct classifications were about 96 for GM and 97 for the ILDs. The study has demonstrated that accurate and reliable pelvic skeletal sex estimation methods are applicable to individuals of younger ages than previously suggested, particularly for the metric analyses or the ILDs. The DFA for ILD and GM both surpassed the 85% accuracy threshold for adult methodologies, and the 75% accuracy threshold that has been suggested for subadult methodologies recommended in forensic anthropology. Therefore, skeletal sex can be estimated when a complete innominate is present that is generally beginning around the age of 10 to 12 years. Standard ILDs defined in the forensic anthropological literature with some slight landmark modifications can be used on individuals with a complete innominate. These findings corroborate the morphological results that accurate sex estimation can be applied at earlier ages. The only requirement for a forensic anthropologist to assess the skeletal sex of unknown remains using either of these methods is a complete innominate. Future research directions will include the study of a large number of individuals spanning the entire ontogenetic period and through adulthood to identify the patterns in growth relating to sex and age. That is the currently funded NIJ research project. Additionally, in future research to improve the methodologies used in forensic anthropology for the identification of human skeletal remains, an ontogenetic perspective will help in assessing the timing of sexually dimorphic features in the pelvis. And future research should also consider the influence of non-hormonal biological factors such as genetics and the effect of hormonal changes throughout the ontogeny of the skeleton. So thank you for your attention and attendance today, and to the NIJ for funding this research. And my email's on the screen now if you have any questions. Thank you.
All right. Thank you very much, Leah. We do have a few minutes for a couple of questions. So I'll start with, there was a large age range in the data set, 8 to 20. Did you see any differences in the accuracy of the sex estimation based on their age? And you did allude to this answer at the end of your presentation, but if you want to give us some additional context, that'd be great.
Yeah. So as I showed in the kind of final results of the two methods, the age of the misclassified individuals was kind of spread throughout that total age range. When I was running the DFAs for the different models, particularly for the ILDs, I did include both the age and the stage of fusion as predictor variables to see if that would affect the accuracy of the methods, and it had no significant impact on the overall estimation. So I ended up removing those as predictors just to keep the data a little bit smaller. But yeah, so no effect that I found in age once you have that fusion of the nominant.
Thank you for that. So our next question is, both the ILD and GM approaches seem to perform well. Is there a method that is preferred in application for forensic anthropologists? And I'll add to that just a little bit. Are you able to combine the results of both of those methods to get an even better predictor?
So great question. I tend to see the GM analysis as more of a research tool to direct the development of methods. It's because it's a 3D kind of analysis. In practice, it's fairly impractical to use when you're in a medical examiner's office and you have remains. But because the shape changes that we were seeing were also being captured by the metric analysis, I think the application of the ILDs is much more practical for use. And so for that method, they can be used on dry bones. You can take those two-dimensional measurements, apply the discriminant functions, and get your sex estimation that way. So I think the ILD is practical and better for actual use, and the geometric morphometrics kind of guides the research direction.
Okay, so yet another variable that I wasn't necessarily considering is really important. All right, so our next question, and then we'll move on to our next presenter. Is there a threshold, confidence interval, or other statistical statement that could help with this data interpretation to minimize misclassifications?
That's a great question. I think for the DFA analysis, all of the accuracies that I reported were from a holdout sample. So the model is trained using 75% of the data. And then that 25% that was excluded is then tested using the model. So that's all new data points. And that 97% accuracy is based on that new data that the model has not seen before. So that 97% accuracy is the essentially the accuracy and confidence of using that model for each individual.
Thank you very much. We did have a few remaining questions in the chat pod. So if you have questions, please reach out to Leah directly to answer those. So we're going to move on to our next presenter, and that is Kaitlyn from the University of Central Florida. Kaitlyn's presentation is titled Development of a Matrix Match Standard for Toxic Exposure of Hair. So take it away, Kaitlyn.
Hello, hi everyone. Thank you so much for coming and happy Forensics Week, National Forensics Week. My name is Kaitlyn Bonilla, as Tracy said, and I'm from the National Forensic Center for Forensic Science at the University of Central Florida. I'm going to be speaking with you today about the development of a matrix match standard for the toxic exposure of hair and my progress, and the characterization of the material and my multi-elemental spiking. So I need to provide a disclaimer that my project is funded by the National Institute of Justice. So the opinions, findings, and conclusions are those of the authors and do not represent the Department of Justice. So I'm going to give an introduction into my research, the chemistry of hair, laser ablation, and what matrix matching of hair looks like. For the chemistry of hair, hair is mainly composed of keratin in that 65 to 95% range, lipids, 1 to 9%, and 15 to 35% water. And then we're really looking at that less than 1% trace minerals. To develop this material, we had to look into the keratin intramolecular bonds. As we can see on the left-hand side, the binding that it takes into account is the hydrogen bonding. There is also disulfide bridging, as well as ionic bonding and van der Waals interactions. The keratin elementally is composed of carbon, oxygen, nitrogen, hydrogen, and sulfur. And then you can see the trace elements here that are commonly seen in hair, and then you'll see these come up later for my multi-elemental spiking method as well. I also wanted to give a brief introduction to laser ablation of hair and why it's really important. So laser ablation is going to allow for direct sampling at micrometer scale spatial resolution. And laser ablation is really important as it is able to perform this highly sensitive analysis with any sample pretreatment. So we're able to directly sample from the hair. We utilize laser ablation ICP-MS system. We have a J200 LA benchtop LIBS system where we utilize a high-powered laser to then create these fine particles that is sent to the inductively coupled plasma. So utilizing this, we're able to get that sensitivity down to that parts per billion level. And then this is also really important in forensic science as it's able to utilize this limited amount of sample with minimal sample destruction. We're able to get a spot size of anywhere from 50 to 100 microns. And as we know, as hair grows, the analytes grow along with the hair. So we're able to also get a quantitative chronological record of exposure in a single hair. So what is matrix matching? So a matrix match standard is a calibration material that is going to provide a similar composition to the analytical sample. So in doing so, this minimizes sample matrix effects. And this is really important in analytical chemistry to ensure that we have a similar instrumental response to the standards in the sample. So in doing so, this is going to improve the accuracy, reproducibility, sensitivity, and method validation. So for hair, matrix matching is going to consist of many things, but I wanted to go over some of the known hair reference materials that are utilized in literature at the moment. So as we can see here, there is hair powder that is pressed into a pellet, glass using the NIST series, and then dogfish liver as well. So as you can see that these reference materials, as they might chemically represent hair, they do not physically represent hair. So for matrix matching, we want a material that's going to have the same chemical composition and physical composition of our matrix. So in doing so, we're developing a material that has a similar matrix composition for chemical similarity, which is going to be composed of keratin. And that is what our material is composed of. And then we also are going to have similar morphological properties for comparable ablation. And this is important. We're going to look at this in our dimensions and our thickness and mass density. And then we also want a sample that is homogeneous at the scale of the sampling technique. So we're also going to examine the homogeneity as well. So our objectives for this experiment, due to the lack of these matrix match reference materials for hair analysis, we're developing a material that is going to emulate the chemical and physical properties of hair for its toxic exposure. So we developed a material that is composed of a keratin film, and I'll explain the synthesis to that in a second, but we are spiking this material and then comparing that to doped single human hairs for its elemental analysis and then molecular analysis later on as well. So for my material, I'm developing it from cut human hairs that are gathered from local hair salons, and the hair is cleaned utilizing a chloroform methanol solution, and that is for 48 hours. And then the keratin is extracted utilizing the Shindai method. And this is composed of, as you can see below, thiourea, urea, beta-mercaptoethanol, and it's incubated at 50 degrees Celsius for 72 hours. And then once we have our keratin extracted, we isolate it using a centrifugal filter at 5,000 RPM for 4 hours. And then this is composed, then we have our keratin solution. So from here, this is where I spike the solution to create our calibration standard. And then the material is cross-linked with trichloroacetic acid and calcium chloride. And then we now have our thin film. So this film is 10 millimeters wide, and it's very thin, and it's very similar to a hair. So I'm going to go over the characterization of our reference material. So we really wanted to characterize the chemical properties and physical properties of the material. So for the chemical properties, we characterized it by looking at SDS page. As you can see here, we have we did extract the alpha keratin at 44 kDa and 58, and we really wanted to ensure we are extracting alpha keratin since we are extracting it and not using keratin powder. So this did show similarity to human hair's alpha keratin content. And then we also are looking at the physical properties, so the thickness of the reference material and compared to human hair. So the reference material was seen to be around 100 microns, and we also can control the thickness of the material, which is really nice because hair is also variable and has different thicknesses depending on someone's hair type. So we're able to control that as well. And then we also looked at the mass density of the film compared to the hair, and then we were seeing that they're also comparable at that 1.3 grams per centimeters cubed. We are also looking into the homogeneity and stability. So for the homogeneity, we want to ensure that the sample is physically and chemically homogeneous at the scale of ablation. So as you can see on the left-hand side in the top image, we did a series of imaging where we looked at the physical homogeneity, and then on the bottom image, we also looked at the homogeneity at the scale of ablation at 1000x. So for the stability, we are examining that over time. We want to ensure that the sample is physically stable and chemically stable. So we're looking at the thickness, the dimensions, the color, and we also want to make sure that there's no shrinkage with the material as well. So now into the fun stuff. So for toxic metal exposure, I'm going to go into my spiking method and the results that I've seen so far. So for my multi-elemental spiking, we developed a tris EDTA buffer. So utilizing this, we wanted to be able to encapsulate and capture that that metal, and then bind it to the protein and also protect the protein. So what we are utilizing is this tris EDTA buffer because we are spiking in 4% nitric acid. So it is composed of ultra pure water, and then we have that spike in 4% nitric, and then we utilize ammonium hydroxide to counteract that spike. We also utilize tris to protect the keratin, and then we utilize EDTA at that 0.075 molar concentration. So as you can see here, we're assuming that the EDTA is hydrogen bonding with the keratin, capturing that metal, and then the tris is there to protect it. So for the multi-elemental spiking, we are developing a series of calibration curves for each element. So these right here are the elements that I spiked with. And then what we did is we spiked it in a series of nominal concentrations, and then we saw the instrumental response, and we correlated the instrumental response to the nominal concentration. So what we are seeing here is a variation of yields. So these are yields that I got back for each correlating element. And we see the yields are anywhere from 11 to 40%. And then for zinc, we have a very high yield. This is due to zinc being present everywhere, unfortunately. So that is something that we are looking into as well. So in conclusion, we're developing this matrix match standard for human hair to be utilized in forensic toxicology. The material has shown to possess similar chemical and physical characteristics to human hair. And then this material can hopefully pave the way for a new set of calibration standards for toxic exposure of hair in forensic investigations. For my future work, there's a lot more to be done. So for elemental analysis, we're still working on the characterization of the material by laser ablation. And then the method will be validated and evaluated by ICP-MS and replicated. We are also going to look into the LOD, LOQ, and linearity. And then another part of the project is the molecular analysis. So we are going to be spiking the material with drug metabolites for MALDI MS analysis. So this will be validated by LCMS and then compared to doped single hairs. So as I stated previously, we're also looking at the stability. So that will be examined chemically and physically over the course of 1 year for the material. So these are all of the parts to the project. So in the acknowledgements, I really want to thank the National Institute of Justice for funding this project and the National Center for Forensic Science, my PI, Dr. Matthew Beaudelet, and my research group. I also want to thank my undergraduate students, Ashley Fox, Chloe Phillips, and Charlene Harris. They've really put a lot of work into this project. And then Dr. John Lucci and Dr. Mara Martinez at the ICANN School of Medicine at Mount Sinai. Please let me know if you have any questions and feel free to contact myself or my PI, Dr. Matthew Beaudelet. Thank you very much.
We do have a few questions that we can cover over the next few minutes. So let's start with, is the matrix essentially a keratin film fiber, or are you able to mimic the structure of hair, the cortex, the cuticle, the medulla, and how that may impact drug retention?
So for right now, it is just a keratin film fiber. I would love to go into the process of being able to replicate all of the parts of the hair. We are looking into incorporating melanin for the drug metabolite study. So hopefully for the drug study, we're in the preliminary stages right now, but we're looking at incorporating the melanin as well.
Good. The questions are coming fast, so we'll move on to our next one. Did you use trace metal grade nitric acid for the spiking, and or did you also test the metal content of the solutions used in the sample prep?
Yes, so we are using the ICP-MS trace metal grade solutions for that. That's why they're in nitric acid. And then we also did test it previously, and that's what I used to create the liquid standards for the ICP-MS to calibrate it, the instrument as well. Thank you for that.
Are the standards only for laser assisted techniques?
So no, right now they are being applied to laser ablation, but the standards can be digested and utilized for liquid techniques as well. But right now we're just utilizing it for laser ablation.
Here we've got a pair of questions, and I'll just start with the first. Can hair reveal exposure to environmental toxins such as pesticides, industrial solvents, or other chemicals present in the air or water, and for how long a period of time?
I would assume so. We are not looking into those, like pesticides or anything, but I would assume so. And then analytes can be present in the hair for up to weeks or months, and even years, just depending on the length of the hair.
Right. Based on the list of new products, how do you reevaluate the list by integrating the product?
I'm sorry, can you repeat that question?
Sure. So it sounds like, so this is the same individual asking, I guess, a follow-on question. So based on the list of new products, so as products become available, how would you reevaluate your list of items that you're testing for? Or do you have any
Depending on
plans?
like the elements. Yeah, so these are all controllable standards, so we can incorporate new elements as they come out or new drugs, depending, the drug binding is going to be a little bit different, but for the elements, we can definitely incorporate them as more studies come out and more research if they need to be.
Sure. So I have a couple more questions to answer, to ask, but I'm gonna just sneak in here with one of my own. So Kaitlyn, actually, we had a chance to talk yesterday when we were at the NIJ conference, and you were telling me all about kind of different applications that you could use this research for. So will you share that with the audience?
Yeah, so it can be used for occupational exposure, medical, clinical, as well. And then I've also had questions about anthropology, but that's more isotopic analysis.
Okay. So next question, why do you use keratin from hair and not just use keratin powder?
And so we extract the keratin because we want to have a matrix as close to hair as possible. And then keratin powder normally is not made from hair. It's normally made from hooves and wool. So that's why we do extract from hairs. And then we purify the keratin to, minimize those interferences.
So if you're using donated hair to make your material from, I guess lot or from batch to batch, are you testing for similarities? Because of course the same individuals are not donating each of those times.
Yeah, exactly. So we definitely look at that. I always make the materials, the calibration standards from the same batch of keratin. And that's something that definitely we are aware of, and we ensure that when we're doing our analysis.
Understood. So we've got a few more minutes and we'll ask this next question and then we'll move on to our next presenter. So how important is melanin to the standard and will it be incorporated? I think you alluded to, the hopes to do this, but maybe tell us a little bit more.
Yeah, so for the toxic metals, we're not looking at incorporating it. For the drug metabolites. It's really important because melanin can aids in the binding of the drug to the hair. So that's why we're looking at incorporating it. So hopefully, like I said, I'm still in the preliminary stages of that, but hopefully we can really get that to work and be able to use that for the drug study.
All right. Thank you very much, Kaitlyn. And what we're going to do now is move on to our next presenter. We're going to hear from Daphne Patten from Iowa State University. Daphne's presentation will cover investigating physical activity from fingerprint lipids using machine learning. Take it away, Daphne.
Wonderful. Thank you, everyone, for joining me today. I'm very excited to share my research on investigating physical activity using fingerprints. I am Daphne. I'm a PhD student at Iowa State University working in Dr. Youngjin Lee's lab. So let's get into it. Yeah, there we go. All right, so just some brief background for everyone. I'm sure many of you know what fingerprints are, but for those of you who might not, fingerprints are an oily residue that's left behind when our hands come in contact with the surface. So there's more than just oils and lipids within fingerprints, but for our purposes, we are mostly interested in these sebaceous compounds and what they can tell us. Now, typically, fingerprints are used to link a person to a location, so a suspect to a crime scene, and they're great at doing this. However, it does rely on being able to match a fingerprint in a fingerprint database. The majority of people, though, are not going to be in a searchable database unless they have an occupation that requires them to get fingerprinted, or they've committed a crime before and are already in the database. And when we run into these issues where a fingerprint does not have a match, it leads investigators with very little information and use for this fingerprint until someone is found. So we thought, what other information can we gain from a fingerprint? Now, our answer might surprise you. We said physical activity. Physical activity is in reference to a lifestyle of a person. So whether someone is active or not can be dictated by their habits, like do they go to the gym or do they like to binge Netflix at home? But it can also reference their type of occupation. So an athlete or a construction worker will have a higher physical activity than a desk worker or a student. So having such empirical lifestyle information can really help investigators narrow their focus and time more efficiently. In the past, we've actually started this research on a smaller scale study looking at triacylglycerols, or TGs as they're abbreviated. And what we saw was in men, we were able to tell the difference between active and not active men, but this was not seen in females. So what we're doing in my research is trying to take a step further and say, why don't we increase the amount of subjects we have and also look at more lipids to see if we can tell the difference in both sexes. So I am actually very glad I go after Kaitlyn, because I'm going to be telling everyone about MALDI, or matrix-assisted laser desorption ionization mass spectrometry, if you want to use the full name. Now, this is going to be slightly different than the matrix that Kaitlyn was presented on, and I'll get into that in my next slide. But what's really great about MALDI is it allows us to analyze solid samples without an extraction process. And this makes it high throughput and still has the same sensitivity that you'd expect with mass spectrometry. So how it works is if we have a fingerprint on a glass slide here, we irradiate it with our laser, and this creates a desorption event, which is a softer ablation event. These ions that form can then be used with our mass spectrometer to analyze all the different compounds found in a fingerprint. This here is a representation of what the sample spectra might look like. And the region in green represents our wax esters, or WEs, as we're going to be calling that. And then we also have our triacylglycerols, or TGs, in the blue region. So just as a general overview of how we analyze fingerprints, what we ask is our participants to give a groomed fingerprint, which is just asking them to swipe their thumb across their forehead and then press it into a cleaned glass slide. This is non-invasive, and usually our participants have more issues with the 8-page physical activity survey that we give them. So it's lovely for the IRB. We then take it back to the lab and we apply sodium acetate onto our glass slide to promote sodium adduct formation. And we follow that with an application of a thin gold film. And this gold film works as our matrix. And for MALDI, a matrix allows us to softly ionize the chemicals so we don't break down our fingerprint lipids with the laser intensity. We then look at the ions created with our Q-exactive Orbitrap MS in positive ion mode. And the signals that we get, we take and normalize to our sum saturated. So the specific lipids would be TG48.1 or wax ester 36.1. And that's divided by the sum saturated TG region. And that's because sometimes people will have different quantities of lipids on their finger at the same time. And it's really nice to be able to normalize to something within their fingerprint already. And that accounts for different variation of deposition. So our next part of our approach is considered the machine learning analysis. So this is where we take our lipids that we just got from the chemical analysis, and we combine that with our physical activity survey. So we did not make the survey. It's 8 pages long, and it was made by the authors listed below in 2010. And it allows us to get a physical activity energy expenditure per weight, or PAEEW. And that is a numeric value that sort of relates to someone's physical activity and allows us to put both that information along with the lipids into MATLAB and conduct machine learning. Now, the next thing you might be asking is, why are we using machine learning? Well, the simple answer is our data is very complex. And while humans can have a hard time finding trends when the data gets too complex, computers excel at this. But they do have to be trained to do this. So in machine learning, you have a training process where it takes an algorithm, or a model as it's more commonly called, and it trains that to detect the differences in your inputted data. We then have a second data set that has been set aside that the model has not seen, has not been trained on, and that's used to test it. And this gives us an objective overview of how well our model is actually performing with data that it hasn't been trained on. For those of you who might know machine learning, we're specifically using a supervised machine learning approach with an ensemble model. Now, just to go over an overview of what all of our steps are in machine learning, these first three boxes in blue are going to be our chemical analysis. So this is what you'd expect from a multi-sample prep. And then in purple here is where we start getting some user input. So we start with the data processing step, where we do any quality filtering that we need to do on the data in case there's contamination within the fingerprints. And we also have our normalization techniques if we are applying them. The data is then split into our training and testing set, with the majority staying in our training. And this is because we really want the model to be trained very well and have a robust algorithm. This is done 10 times to minimize sample bias just from the random sampling, and also to form statistics. The next part is our training set is used in order to train a model and optimize the hyperparameters. So hyperparameters are simply settings that you can have in the algorithm that have to be optimized for every machine learning attempt. We then get a trained model that can go into the testing, and then it enters the evaluation stage. So at the evaluation stage, we look at both the testing and the training, and we ask, is the average accuracy greater than or equal to 80? Now, this is a value that we set ourselves because even 80% is a low bar for forensic investigations, but oftentimes it's a good start for when you're starting machine learning. So that's the bare minimum we were comfortable with. If the model does not meet this 80%, it is considered rejected, and we go back to the beginning here and we look at changing the algorithm, or we change the settings of the algorithm, and we continue this loop until it is accepted. I will be presenting on 2 different models coming up, but first I need to go over one other factor to keep in mind with my data. And that's the presence of what's called an inconclusive margin, or a gray zone. So these are common in biological studies, and it's used to help with binary classifications of very homogeneous data that we often see in human samples. So what it means is, in this first model I'll be showing, we have our inactive class and we have our active class, and in between is a gray buffer zone. So anyone that falls in the small zone are actually going to be removed before machine learning, because our model just cannot tell the difference between people in this area, and it causes a lot of issues when it comes to the accuracy of the model. This does have the caveat, though, of not having a continuous score of physical activity with this gap area. So it decreases the applicability across all physical activities. Luckily, our second model does not suffer from this same necessity, and it has a continuous spectrum across inactive and active categories. So keep that in mind when we get to the results. All right, so the next thing I need to talk about is how we evaluate our machine learning models. So the first metric that I'll discuss is the accuracy, which is pretty straightforward. We have how well our model's actually doing in a percentage of an average in the 10 replicates and the standard deviation within the parentheses beside it. Ideally, we want this at 100%, but life's not ideal, so it will be less than that. The second metric we have is called a confusion matrix. And what this shows is how our model is actually classifying the samples it's handling. So on the side here, we have our true classes, and above we have our predicted classes. So our hope is anyone who is active will be classified as active, and they will fall within this blue box here with the star. Same can be said for the inactive. We still want it in the blue box. The orange boxes represent our false classifications, and I want to draw attention specifically to our false positives. So that's someone who's inactive that's being classified as active. And in a forensic sense, this is what we really want to avoid, because the number one rule is we want to give correct information, not incorrect. So less information is better than incorrect. And that will just make sure that we don't send investigators on incorrect wild goose chases. The next characteristic that we're going to cover is the receiver operator characteristic, or the ROC curves. Now, these take a bit more to get used to, but on the y-axis, we have our true positive rate. So this is the rate of how many classifications are correct. And on the x-axis, we have false positive rate. So this is how many are being incorrectly classified. So we want this ideally to be 0, 1 on the scale, right where this star is. Again, that's an ideal, but when it does match up like this, what we'll see is an area under the curve of about 1. Now, this is a great metric in order to be able to tell how well your model is actually performing. If the model has an AUC of about 0.5, you'll find it closer to this dashed line here, and that represents more random chance or a coin flip. And since we're using computers to make these predictions, that's considered very bad for machine learning. All right, now the moment we've all been waiting for, to get into some of our results. So this first model is our triacylglycerol model. It uses a log scale transformation, and importantly, sex and biological age are included, as well as a gray zone. Excitingly, though, we do see a train-in and test-in accuracy of about 90%, and this is reflected in our confusion matrix. So we see the majority of samples are being classified within the blue boxes, which is a correct classification, and that holds true for the test-in as well. One of the things to note, though, we did see more of the false negatives, within our train-in set. So this means some active individuals are being classified as inactive, which is not desirable, but it's a better problem to have than false positives. And this is reflected well in our ROC curves. You can see here that we have average AUCs of about 0.9, especially in the test-in data, which again, that's the data that the model was not trained on. And one thing I'd like to point out, though, is the deviation that we see in the averages are a bit more than I would like. So that's one of our goals with the next model, was to try to improve this deviation that we see. In the next model, we combined both triacylglycerols and WEX esters in an attempt to improve our machine learning results. And importantly, we were able to get rid of that gray zone. So that makes this a lot more applicable. The same sex and biological age was included as factors in the machine learning. And we see an average of slightly less than 90, but within the same error margins as our last model. And this can be seen in our confusion matrix again. So we have majority being classified in our correct classifications, but I will say we do have an increase in our false positives, just slightly, in both the train-in and the test-in. So this isn't what we want to see, but if we look at our ROC curves, we do see about a 0.9 still as our average AUC for both train-in and test-in. And in fact, the variation is less now in this model than we saw in the last one. So roughly the two models have about the same metrics, but with the difference of the applicability in physical activity. So in conclusion, we have two machine learning models that can predict physical activity with about a 90% accuracy. The difference is the first one uses fewer lipids and has a limited applicability because of the inconclusive margin, while the second one does require more lipids, but it can be used across all physical activity ranges. In the future, what we plan to do is continue improving, especially the second model. And we plan to do that using feature selection, which is the term we use to tell we're going to look at all of our lipids and see if we can remove some that are less important than others. And this can help just improve the accuracy and the metrics of the machine learning just by little amounts, which do matter. The other thing that we're planning to try is more normalization techniques. Our second model does not have any normalization on it currently. And the first one did improve when we added a log scale transformation. So, and lastly, we hope to prepare and submit a manuscript on this. All right, I would like to thank everyone for their attention and the NIJ for this wonderful opportunity and their funding throughout my graduate student career, and my colleagues in the Youngjin Lee lab and my mentor, Dr. Youngjin Lee. And I'd love to answer any questions we have.
All right, thank you very much, Daphne. So I'm going to move on to questions. So our first question is, has there been any research into common forensic fingerprint powders that can be used in conjunction with MALDI?
Yes, so that's a great question. Our lab has done a lot of work when it comes to being able to see if we can compare forensic common practices with our MALDI techniques. And what we found is carbon black fingerprint powder, one of the most common fingerprint powders, is actually a really good matrix for our analysis. And so you can actually use that instead of gold. For our purposes, we still use gold because in general, it's a cleaner matrix than the carbon black fingerprint powder, but you get good signal with either.
Thank you very much for that. So my next question is that you've compared active and non-active individuals, but have you also looked to determine the difference between male and female based on TG content? Are they also distinguishable?
Yeah, so that's a great question. We haven't done much work into see if we can just use the lipids to tell the difference between male and female. So it does seem from our initial research that there is a difference, but we're not sure exactly what that would be. And I do know that there's other research that looks into amino acids within fingerprints that can tell the difference between sexes like that.
Fairly exciting. All right, so our next question, and then we'll see if we have time for one more. We've got a couple in the pod. How does the activity level questionnaire classify individuals in a sedentary occupation, but are also into fitness? Would they be considered active or non-active?
Yeah, that's a great question. So the questionnaire actually has many parts to it, and it looks at many parts of a person's life. So what we get is a numerical value, and that can change based on if someone does have sedentary or if they're very active in the gym. So there will be some overlap. So if someone does work a desk job, but they go to the gym for 2 hours a day, we're not going to be able to tell if they get that activity from their own personal habits or if they get that from their occupation. So that is a bit unclear, and investigators would have to do a bit of legwork when it comes to that. But they would know that the person is physically active.
Understood. All right. And that helps us understand, on the back end, how that data might be then used by investigators as well. So with that, we'll conclude. Thank you very much, Daphne. And we're ready to move on to the next presenter. So up next, we're going to hear from Ciara Rhodes from Virginia Commonwealth University. So Ciara will be giving us a presentation on optimized demineralization methods using bone slices for improved DNA recovery. So over to you, Ciara. Thanks.
Thanks, Tracy. So today I'll be discussing my dissertation research with you all, and I'm very excited. But first, I just have a couple of quick disclaimers that I need to state. So this work was supported by 2 grants, and the opinions, findings, and conclusions are our own and do not necessarily reflect those of the Department of Justice or the Forensic Sciences Foundation. Additionally, we are not affiliated with, nor did we receive funding from any of the commercial suppliers of the reagents and equipment used within this project. So forensic analysis of skeletal remains is typically undertaken after a mass disaster incident or a missing persons investigation where skeletal remains may be the only biological sample that's available for human identification. This was the case after the 9/11 terrorist attacks on the World Trade Center, and the skeletal remains recovered were likely exposed to further degradation due to fire or chemicals, which implicated the ability to obtain genetic information for identification purposes. And even when skeletal samples are in relatively good condition, these are some of the most challenging samples to process due to the structure of bone. Bone is a composite material that provides stability and preservation to endogenous DNA molecules, and the calcified matrix itself presents a physical barrier to the isolation of DNA from skeletal elements. Traditionally, samples are processed using pulverization, and this is typically performed using liquid nitrogen coupled with a mortar and pestle and mechanical force to grind the bone into a fine powder. It's a highly destructive process that consumes potentially limited samples, and most published protocols require 0.2 to 2.5 grams of starting material for DNA isolation. Pulverizing the bone into a fine powder enables the release of the DNA molecules from the hydroxyapatite mineral matrix, and a demineralization pretreatment may be performed on the powder prior to DNA isolation. Using EDTA, the main mechanism of demineralization is to inactivate DNases by chelating calcium and magnesium ions, and this aids in the breakdown of those mineralized complexes. However, the co-isolated intrinsic components of bone, such as calcium ions and collagen, as well as any exogenous DNA from microbial infiltration, can impact the success of the endogenous template DNA amplification during PCR. For this work, we wanted to investigate alternative front-end processing methods that could minimize potentially process-induced degradation of the template DNA and provide improved allele recovery and higher quality STR profiles from degraded skeletal samples. So first, we needed to select an organism that was most representative of humans, and this was determined at the collagen level. We performed evolutionary analyses of several domesticated animal species using MegaX software to infer the phylogenetic relationship of 2 collagen gene transcripts. While we found that dog followed by cat shared the closest relationship to human, cow was the 3rd most similar, and given its dimensions and the relative ease of obtaining aged and weathered skeletal cow remains, we selected cow for this work. So these are the bovine samples that we used for our initial testing, and it was cortical bones from 2 individuals. The bovine samples were collected from aged and weathered skeletonized remains that were found on rule properties, and no animals were harmed to obtain these samples. Per the livestock owners, the animals expired naturally and decomposed in pastures or woodlands of the southeast region of the United States for more than 5 years before collection, and the skeletal remains were exposed to uncontrolled environmental conditions. For sample preparation in a dedicated chemical fume hood for bone processing, the samples were cut following the traditional method, in which the exterior surface area of the sampling site is sanded with a rotary tool, and a small window of bone from the midshaft region is then cut using a rotary tool with a diamond cutting wheel. After cutting, the bone pieces were placed into sterile conical tubes, and they were soaked in a 50% bleach solution for 5 minutes. Following that, they were rinsed with deionized water until the water was clear and no longer retained the smell of bleach. And then a final rinse of 100% ethanol was performed for allowing the samples to dry in a sterile biosafety cabinet overnight. We also wanted to conduct a pulverization comparison study. So a comparative analysis was performed using the traditional processing method of pulverization with organic DNA extraction. The bovine cortical bone samples were powdered using a sterile mortar and pestle with liquid nitrogen, and each bone element was sampled in triplicate, and they were 0.1 grams of starting material. The powdered samples underwent a digestion where they were incubated in an extraction buffer with protease for 4 hours at 56 degrees prior to beginning the organic DNA extraction, followed by ethanol precipitation purification. In addition, a silica-based extraction chemistry was evaluated on an additional subset of pulverized samples, and we optimized this using the ChIAMP DNA blood mini kit. Assessing both organic and silica-based extraction chemistries within the pulverized samples was important, as silica was utilized as the extraction chemistry for the demineralized slices. For the total demineralization of the bone pieces, the samples were added to sterile 5 milliliter tubes with 4.5 milliliters of demineralization buffer. The tubes were then placed into a tube revolver, which was then housed inside of an incubator set at 56 degrees Celsius, and the samples were allowed to demineralize for 3 to 5 days, with the buffer changed every 24 hours. Following the demineralization process, the samples were soft and easily able to be sliced. I'd also like to note that fixation or embedding of the samples was not performed before slice preparation. They were easily sliced with a sterile razor blade, and we created biological triplicates of 0.1 grams of bone slices, and these were obtained for all of the bone samples. They were then stored at minus 20 degrees Celsius until DNA extraction. Following slicing, we performed silica-based extraction chemistry on both the pulverized samples subset and the slices. We utilized a large volume lysis in conjunction with Appendix I from the DNA Mini Blood Mini Handbook for crude cell lysates and other samples, in conjunction with the DNA purification from blood or body fluid spin protocol. So for the bovine samples, the forensic DNA workflow is shown as follows. After DNA extraction, the samples were evaluated using the Tribo bovine qPCR kit to assess the amount of total bovine mitochondrial DNA within the samples. And we were also curious to see how much exogenous DNA was actually within these samples. And so we looked at the microbial DNA qPCR assay as described by Seashels-Williams for 16S ribosomal DNA. Following that, we performed DNA amplification using the Thermo Scientific Bovine Genotypes Panel 1.2. This panel encompasses the 12 STR loci that are recommended by the International Society for Animal Genetics for routine use in bovine parentage testing and identification. Following amplification, the samples underwent capillary electrophoresis and STR analysis with the 3500 genetic analyzer, and electropherograms were analyzed with GeneMapper IDX software. We had an analytical threshold of 150 relative fluorescent units. So for the bovine quantitation results, they're shown here. On the left side of the screen is the bovine mitochondrial DNA, average total DNA yields for each of the treatment groups. And on the right is the microbial 16S ribosomal DNA, average total DNA yield within each of the treatment groups. There were 15 observations for each treatment group as the samples were tested in triplicate, and we had 5 bones. And there were no statistically significant differences between both of these quantitation results. So moving forward and further our analysis of the DNA integrity through STR genotyping, this is an electropherogram of an overlay of all of the channels for both the pulverized sample and demineralized slices sample for bovine femur C in triplicate. And on the left, we have the pulverized samples, and on the right, we have the demineralized slices. Demineralized slices showed less degradation or ski slope effect within the larger base pair loci, and it had better quality STR profiles. Bovine profiles generated from demineralized slices yielded higher average percentage of alleles detected, and it also demonstrated greater retention of STR loci than the pulverized samples. Therefore, using demineralized slices resulted in more complete, balanced STR profiles with higher peak heights and less degradation than the powdered samples when they were both having a DNA input concentration of 0.5 nanograms. So following the development and optimization of our prolonged demineralization and slicing method with the bovine samples, we wanted to test our method's efficacy using degraded human skeletal samples. So those processing methods, both pulverization versus slices, were utilized with our silica-based extraction chemistry, and we evaluated the endogenous and exogenous DNA amounts within these, and also utilized link-based and targeted sequencing technologies. So for the human skeletal samples, they're displayed here. There were cortical bone, both left and right femurs, from 7 individuals. These were Virginia police cases from the 70s to the 90s that were brought to the state's Office of the Chief Medical Examiner. Once they were deemed historical and of no use for forensic DNA testing, the custody of the samples was transferred to forensic anthropologist Dr. Tal Simmons for teaching purposes. The samples are estimated to be 19th century human skeletal remains, and as you can see, these samples were subjected to uncontrolled environmental conditions and/or scavenging prior to collection. So for the human samples DNA workflow, the skeletal samples underwent the same sample preparation and cleaning routine as the bovine samples before pulverization or prolonged demineralization with the subsequent slicing. Pulverized and demineralized slice samples were prepared in triplicate of 0.2 grams for each skeletal element. Following DNA extraction, the samples were quantified using quantifier trio for the assessment of total human and human male DNA. And again, we utilize the 16S ribosomal DNA qPCR assay as described by C. Schultz-Williams to evaluate the amount of exogenous DNA within our samples. For DNA amplification, samples were amplified with the PowerPlex Fusion 6C system, and we increased the amount of cycles of PCR to 30 instead of the 29 manufacturer's recommendation because of the quality of these samples. After amplification, the samples underwent post-amplification purification with the QIAquick PCR purification kit, and then they were assessed with capillary electrophoresis with the 3500 genetic analyzer, and we increased the injection time to 25 seconds, and the STR profiles were analyzed using GeneMapper IDX software. So for the human samples quantitation results, they're shown here. On the left side, we have the average human DNA yields for both of the treatment groups. We also performed a inhibition assessment utilizing the IPC-CT values of our samples. For the demineralized slice sample set, we had 73% of the samples that had passing scores for the IPC assessment, and for the pulverized samples, we had 52%. I'd also like to note that the average degradation index for the pulverized samples could not be calculated due to failed large autosomal amplification. On the right, we have our average bacterial DNA yields for both of the treatment groups, and as you can see, when we have a sample that is less processed, we're getting more amplifiable DNA with these quant values, as you can see. And then it's very contrasted when you look at the pulverized treatment group in comparison to the slices, whereas we have a lot of the samples in their triplicates were undetermined with their CT values. So for the human samples STR genotyping success, this is again showing an overlay of all of the dye channels of the electropherograms for samples 82871 and 1971 replicates. On the left, we have the pulverized samples, and on the right were the slices. No pulverized samples yielded STR profiles, whereas demineralized slices of the same donors yielded partial profiles and ranged from 6 to 34 called alleles across the donor replicates. As you can see, larger base pair length loci are less retained. However, this is a known limitation when working with highly degraded historical samples. So in conclusion, partial STR profiles for 2 of our 7 donors were generated from demineralized bone slices. And the demineralized slices ultimately resulted in greater recovery and amplifiable DNA than the traditional homogenization methods within these 19th century human skeletal remains. Our future work will be taking our DNA extracts from the remaining sample replicates and evaluating them with high throughput sequencing for SNPs and mitochondrial DNA. Utilizing targeted based sequencing may provide additional genetic information and insight from these highly degraded human skeletal samples. So I'd like to acknowledge the National Institute of Justice and the Forensic Sciences Foundation for funding this work. And I'd also like to give a special thanks to the Forensic Technology Center of Excellence for allowing me this platform to share my work with you all today. The VCU Department of Forensic Science, especially my mentor, Dr. Sarah C. Schultz-Williams, and members of the Dawson, Green, and Williams lab. And thank you for your attention, and feel free to reach out to me if you need to ask any questions.
Thank you very much, Ciara. All right, so we do have a few minutes for questions, and we'll start there. So the first question is, based on your results, do you think additional time added to the front end of the workflow, 3 to 5 days for the prolonged demineralization before slicing, is a fair trade-off, and why?
Yes, I think I do believe that it is a fair trade-off, because within the demineralized slice samples, we were actually able to get more information about them at the quantitation level. We were able to get both large and small and Y autosomal target values for those samples, versus the pulverized, we only got small autosomal target values. And so we actually could calculate a degradation index for each of the samples that did have both large and small autosomal values. And this also gave us more insight about the integrity and quality of the DNA prior to moving forward with our STR genotyping, and then influenced our decision to also incorporate targeted based sequencing because of the quality of these samples.
Understood. So we have one more question, and then I have a few of my own. So,
Okay.
What if cortical or compact bone isn't available? Would this method work with trabecular or spongy bone?
So we chose cortical bone because traditionally long weight-bearing bones are what are typically sampled for forensic DNA analysis of skeletal remains. However, it would be very interesting to try to test trabecular or spongy bone because it's more porous and not as dense as cortical bone. I believe that it probably could take less time during the incubation for the demineralization process and the subsequent slicing. However, caution probably should be taken, especially if it's a sample that has been in the elements and have the potential to have microbial infiltration, as the trabecular bone can harbor detritus and other unwanted components. And it's typically usually sanded away when people are sampling before extraction. So it'd be interesting to see if it does hold true and have similar results as these data.
Thank you. So, full disclosure, as someone who has sanded a whole lot of bones in my past practitioner years, this is a significant change to a protocol that's been used historically, for a number of years. What do you think represents the success rate of using slices over using a historical protocol that's been in place for a number of years?
I was surprisingly shocked to actually obtain data from this work with these human samples, especially considering the quality that they were in when I received them. I think that the results from this study could definitely be taken forward and maybe even tested with ancient DNA type samples, and maybe that could increase the amount of information that can be recovered about those samples when in comparison, if you were just to utilize the tried and true traditional method. I think that it is exciting to see more and newer methods brought about into the literature. And so stay tuned because this will be submitted for publication very soon.
This is really exciting work and has the potential to absolutely change, the human identification process for bone material. And with that, we're out of time for Ciara, and we're going to move on to our last speaker of the day. So our final presenter of the day is Neil Zhang from the University of Rochester. So Neil's presentation is on generalizing audio deepfake detection for forensic and security. So without further delay, I'll pass it off to Neil.
Thank you for the introduction, Tracy. Hello, everyone. I'm Neil. So I'm currently a final year PhD candidate at the University of Rochester. So my topic today is generalizing audio deepfake detection for forensics and security. So among this title, the word deepfake might sound not familiar to many of you. So deepfake actually means using deep learning technology. So deep learning is part of the current AI technology that use deep neural networks to train based on some data. So deep learning can be very powerful and used to create some fake media. So the materials I study is on audio. So I study audio deepfake detection. Today's outline will be talking about many of the audio deepfake detections, including speech deepfake, singing voice deepfake, and audiovisual deepfake, and also some techniques about audio watermarking. So I'm very excited to get started. First, some backgrounds. So recently we can see many of these news that these AI techniques can be used to create this audio deepfakes and cause some crimes. To me, this is a very new type of crime. And it only happens like, I think the first one I saw was back in 2019. And we can also see many of these came very recently, like the cloned voice are used in kidnapping or like stole someone's money, trying to pass through the voice biometrics and get verified, okay, then the money is stolen. And also this can be abused to like mimic celebrities' voice and spread misinformation. So all of these are happening recently, and also that's why the audio deepfake detection are being increasingly more important in recent days. And I'll show you some demos of the current speech deepfakes, like how does this sounds, and you can have a feel about like, does this sounds real to you? So the first one,
which he kept concealed in a hiding place with a trap door just under his bed.
Okay, and then let's hear the third.
They swarmed up in front of Sherburne's palings as thick as they could jam together, and you couldn't hear yourself think for the noise.
So these are speech deepfakes, and they are created using either text-to-speech or voice conversion. So usually either text information are going through acoustic model to give you the mouth spectrogram, or the one speaker's voice is converted to the mouth spectrogram, and then use the vocoders to reconstruct the audio. So to me, all of this sounds quite realistic, and I cannot tell the difference whether it is real or fake. And from a survey conducted by this AI company called VoiceBot, many of their customers are very concerned about this deepfake technology. The top one shows the video deepfakes, and the bottom one shows the voice deepfakes. So some more background about how this become into a research topic. So many years ago, people started to study voice biometrics. They want to use voice to verify the person's identity. And at that time, people are developing a system called automatic speaker verification, where we expect the inputs for this verification system is bona fide speech, and we can make a decision whether to accept this person's identity or reject it. However, when the deepfake comes, they can also pass the speaker verification. So this assumption does not hold true anymore. That's why we need another system. We call it deepfake detection system or anti-spoofing system to detect whether the input is from real human or not. So the whole pipeline is like this. So we have some audio recording, and if the speech are from text-to-speech or voice conversion algorithms, then we feed that into the deepfake detection model, which has the front-end feature extraction or the back-end classifier to classify whether it's real or fake. Only when it's real, we pass it into the speaker verification system to get the result of whether this belongs to a target speaker or non-target speaker. So the research questions for the speech deepfake detection contains these three. What features are useful? What models are best to do this backend classification? And also the most important research question to me is like, how can we defend against unseen deepfakes? Because when we develop machine learning models in the training set, we only have we only are exposed to some of the deepfakes we already know. But when we want to deploy the system, we need to generalize to unseen deepfakes. So existing research in many of previous years, people study features and backend models, and my research falls into the third one, training strategy. So the first research question I study is to try to generalize the speech deepfake detection model to unseen deepfakes. So the motivation is that the development of the speech synthesis models are quite fast, and the unseen deepfakes are becoming increasingly more threatful. And we want the anti-spoofing system or speech deepfake detection system to be better trained so that we can defend against unseen spoofing attacks. In other words, we want more generalization ability. So to generalize, one method I propose is called this one-class learning. So the main idea is to compact the representation of bona fide speech while we want to isolate the speech defects or spoofing attacks away from the bona fide speech. So during training, we encourage the latent space or the embedding space of the model to push the speech features like this so that we can build a tight boundary around the bona fide speech and push away the deepfakes. So this is the loss function OC submax I proposed. And from the results, when we compare traditional binary classification methods that use loss functions such as softmax and AM softmax, which represents additive margin softmax, our OC softmax have better results especially on unseen attacks, which contains in the evaluation set of the data set. And from the visualization, we also see that during evaluation, we have some unknown distributions which are quite different from what we have seen in the training and validation set. And also, this PCA plot shows that because the PCA has linear property, then it shows that the embedding space formulates as what we expect. And this achieved the state-of-the-art performance at that time. And there have been many of the follow-up works in the community that further developed this one-class learning-based deepfake detection methods and improved and also generalized that to some other scenarios. And in our group, we also extend this work and propose further one-class learning loss functions. And also one of them is using speaker attractor multi-center one-class learning, which the general idea is to, instead of cluster the bona fide speech into just around one center, we also incorporate the speaker information so that the bona fide speech can be incorporated as multiple centers, and the spoofing attacks can be then separated away. You can see that these triangles, previously they are inside the bona fide cluster, but then if we model the speaker information, it can be further separated. So with this proposed SAML algorithm, we further improved the generalization ability of the Bonilla one-class learning. And the next research question I dived into is try to generalize to more diverse bona fide speech, because in the previous data sets, all the bona fide speech are lab recorded, and all the spoofing attacks are generated based on that lab recorded speech. But when we want to use this system in real world, we know that the audio recording are quite noisy, or they are from everywhere. So the main difference we identify are these audio effects that's caused by the recording and transmission process, and we call this channel effects. And we propose some methods, including data augmentation and some training strategies to improve the channel robustness of the audio deepfake detection methods. Further details are included in this inter-speech paper. And then we have moved on from speech deepfake detection to singing voice deepfake detection. And this is also kind of a very recent technique that makes the singing voice sounds very naturalistic. I'll play this demo for a few seconds. Yes, so it's showing the AI Taylor Swift are covering a sound that's not sung by her in any of the previous times. And we create a database called Singfake, which aims to develop this new research area, singing voice deepfake detection. And in this data set, we collect data from the existing public video platforms, and we include many of the pop songs from different countries and language. And this is the distribution of our data sets. We have each slice represents a speaker, and we have multiple sets to test different scenarios. And we also include a controlled version, which due to time, I'll skip this part. And further, we generalize our one-class learning methods to audiovisual that improves both the audio modality and the visual modality in the video deepfake detection scenario. And last but not least, the work we are currently working on is to build a watermarking system so that we don't need to just do classification for audio deepfake detection, but also we want to add the watermark into the generation system so that all the deepfakes generated by this system will be watermarked. And then when people want to use this fake data, we can easily build a watermark detector to detect whether the watermark is in that audio or not. And for future research, we are extending that to more diversified scenarios of deepfake attacks. And also we want to improve the robustness and also the interpretability for audio deepfake detection system. And for references, these are all the papers we have published along this main theme of audio deepfake detection. And I want to thank all the co-authors from our lab. And also the highlighted papers are what we have discussed in today's presentation. Okay, so these are the takeaway message. So thank you, everyone. And I'm very happy to take any questions.
Right. Thank you very much, Neil. Well, we do have time for a few questions before we close out this afternoon's session. So we'll start with the first one. So how much voice data is needed to clone a person's voice?
Well, that's a very good question. So as techniques evolves, so like in many of decades ago, we may need like several hours of data, but like several years ago, we may only need several minutes of data. And from very recent news, which I shared in the first few slides, you can see that Microsoft developed a voice engine that can clone anyone's voice from just 3 seconds of sample, which sounds very scary. And recently, this is at the beginning of 2023. I believe recently people can even achieve like shorter period of time that you can just clone people's voice with that.
Understood. That is quite a change over the years.
Yeah.
So we have one more question so far. So automated speech recognition. So automating speech analysis processes helps us identify anomalies in pitch, frequency, and unnatural pauses. Up to what acoustic frequency?
So the frequency, usually when people look at speech, we look at the sampling rate of 16K. So that is the frequency from 0 to 8K hertz frequency bands. So most of the time, these frequency bands cover speech very well. And most of the existing automatic speech recognition system or any speech analysis system, including this audio deepfake detection system, are focusing on this frequency band from 0 to 8K. However, if you want to detect more artifacts or want to have more information, maybe some high frequency information that are not included in human understanding of speech. So I believe the frequency bands beyond 8 kilohertz would be still useful. For example, when I do the research on singing voice deepfake detection, because we see some of the artifacts are actually from the music side. And in music, people use the sampling rates of 44 kilohertz or the 48K hertz. So that's more of a sampling rate for music. And we do identify some of the artifacts are from there. So I believe if we use higher sampling rates, maybe it could be also useful for identifying speech deepfakes.
Good. Thank you for that answer. So our final question is this. What challenges do you anticipate for the use of this method in casework or in courtroom testimony?
Sorry, I didn't hear the use case clearly. Could you repeat again?
Sure. What challenges do you anticipate for use of this method in casework and or courtroom testimony?
Courtroom testimony?
Yes. Yes.
I see. So basically, currently, we are mostly developing audio deepfake detection methods based on classification. So by classification, we mean we train machine learning models to try to classify whether this either speech or singing voice or any audio is real or fake. So it's a binary classification problem. So in the court case or some scenarios, is this kind of classification or prediction from the machine learning algorithms are all probability based. So we can only give we believe 90% this belongs to real, or 10% belongs to fake, or 60%. So we can only give this unsure number currently. So it cannot be used as evidence. So that's the limitation of the current method if we want to use this in court case. However, we are also actively developing this audio watermark techniques that once the watermark is injected into the audio and people, and if this watermark is robust to many scenarios, whatever re-recording or transmission, if we can still identify this watermark in this new speech, then we can say, okay, this watermark is robust. Then we can inject some message, if people are using AI techniques to generate this audio, and we inject the watermark saying that, okay, this audio is AI generated, so be careful when you use it. Then I believe once this audio used as evidence in the court scenario, then it can be more served as evidence at that time.
So thank you very much, Neil, for explaining that difference. And with that, I'm going to say thank you very much to everyone for tuning in to our third annual NIJ Forensic Graduate Research Symposium. We'd also like to give a hearty thank you to our presenters for taking time to prepare and provide their presentations today. Unanswered questions should be directed to the presenters. National Forensic Science Week is in full swing, and we encourage everyone to visit the forensic COE or forensiccoe.org for additional activities like the daily forensic science crossword or murder mystery activity. We hope everyone has a great rest of their afternoon, and thanks again.
As you depart, we kindly ask that you take a minute or two to complete our post-event questionnaire. The link will be pasted in the chat and also appear as you exit the Zoom room. Your feedback helps us improve our outreach efforts and continue to provide the best experience possible. Thank you again for attending, and we hope everyone has a great day.